Beyond calories. Study reveals genes responsible for dietary restrictions' lifespan extending effects. Before proceeding further in the video, if you like our video, then subscribe the channel and share it. Scientists have now pinpointed how dietary restrictions not only influence the digestive system, but also significantly impact brain aging, offering potential therapeutic targets for age-related neurodegenerative diseases. Restricting calories is known to improve health and increase lifespan, but much of how it does so remains a mystery, especially with regard to how it protects the brain. Scientists from the Buck Institute for Research on Aging have now uncovered a role for a gene called OXR1 that is necessary for the lifespan extension seen with dietary restrictions and for healthy brain aging, published in the journal Nature Communications. This study was led by Dr. Kenneth Wilson from the Buck Institute for Research on Aging. When people restrict the amount of food that they eat, they typically think it might affect their digestive tract or fat buildup, but not necessarily about how it affects the brain. As it turns out, this is a gene that is important in the brain, explained Dr. Wilson. The study, conducted in fruit flies and human cells, demonstrated a detailed cellular mechanism showcasing how dietary restriction could potentially delay aging and mitigate neurodegenerative diseases. Dr. Pankaj Kapahi, a co-author of the study, emphasized the discovery, saying, "We found a neuron-specific response that mediates the neuroprotection through dietary restriction. Strategies such as intermittent fasting or caloric restriction, which limit nutrients, may enhance levels of this gene to mediate its protective effects. The gene is an important brain resilience factor protecting against and neurological diseases," added Dr. Lisa Ellerby, another senior researcher and co-author of the study. In earlier research, members of this team had shown mechanisms that improve lifespan and health span with dietary restriction. But there is so much variability in response to reduced calories across individuals and different tissues that it is clear there are many yet to be discovered processes in play. Therefore, this project started with the aim to understand why different people respond to diets in different ways. By examining around 100 fly strains with different genetic backgrounds, the research identified 5 genes, 2 of which had counterparts in human genetics. The flies were raised with 2 different diets, either with a normal diet or with dietary restriction, which was only 10% of normal nutrition. The chosen gene, mustard MTD in fruit flies and oxidation resistance 10XR1 in humans and mice, was found to play a critical role in protecting cells from oxidative damage. As it turned out, the gene protects cells from oxidative damage, but the mechanism for how this gene functions remained unclear. The loss of OXR1 in humans results in severe neurological defects and premature death in mice. Extra OXR1 improves survival in a model of amyotrophic lateral sclerosis ALS. In order to identify how a gene that is active in neurons affects overall lifespan, the team did a series of in-depth tests. They found that ORX1 affects a complex called retromer, which is a set of proteins essential for recycling cellular proteins and lipids, and, thus, is a key player in brain aging and neurodegeneration. Retromer dysfunction has been associated with age-related neurodegenerative diseases that are protected by dietary restriction, specifically Alzheimer's and Parkinson's diseases. The team's in-depth tests revealed that MTDAOXR1 preserves retromer function, influencing neuronal health, brain aging, and the extended lifespan observed with dietary restriction. By eating less, individuals may enhance the expression of OXR1, maintaining proper protein sorting within cells. Diet is influencing this gene. By eating less, you are actually enhancing this mechanism of proteins being sorted properly in your cells, because your cells are enhancing the expression of OXR1, explained Dr. Wilson. The researchers further observed that boosting MTD in flies led to an extended lifespan, prompting speculation about a potential link in humans. The next phase of this research involves identifying specific compounds that increase OXR1 levels to delay brain aging, offering insights into the degeneration of the human brain. Hopefully from this we can get more of an idea of why our brains degenerate in the first place. Diet impacts all the processes in your body. I think this work supports efforts to follow a healthy diet, because what you eat is going to affect more than you know, Emphasized
If
Dr.
you like
Wilson.
the video, then like the video, share it and subscribe to Stranger and Zoning 18 channel and press the bell icon. If you